Dopamine may have given humans our social edge over other apes. Compared with other primates, both humans and great apes had elevated levels of serotonin and neuropeptide Y in the basal ganglia. However, in line with another recent study on gene expression, humans had dramatically more dopamine in their striatum than apes, they report today in the Proceedings of the National Academy of Sciences. Humans also had less acetylcholine, a neurochemical linked to dominant and territorial behavior, than gorillas or chimpanzees. The combination is a key difference that sets apart humans from all other species, Raghanti says. Those differences in neurochemistry may have set in motion other evolutionary changes, such as the development of monogamy and language in humans, theorizes Kent State paleoanthropologist Owen Lovejoy, a co-author. He proposes a new neurochemical hypothesis for the origin of hominids, in which females mated more with males who were outgoing, but not too aggressive. And males who cooperated well with other males may have been more successful hunters and scavengers. As human ancestors got better at cooperating, they shared the know-how for making tools and eventually developed language, all in a feedback loop fueled by surging levels of dopamine. Cooperation is addictive, Raghanti says. Lovejoy thinks these neurochemical changes were already in place more than 4.4 million years ago when Ardipithecus ramidus, an early member of the human family, lived in Ethiopia. Compared with chimpanzees, which display large canines when they bare their teeth in aggressive displays, ramidus males had reduced canines. That meant that when they smiled like male humans today, they were likely signaling cooperation, Lovejoy says. Molecular genetic studies suggest that executive function is modulated by dopamine levels in the prefrontal cortex and that different alleles of the catechol O-methyltransferase (COMT) gene are responsible for different cognitive profiles. The COMT gene encodes a catabolic enzyme involved in the degradation of dopamine. A polymorphism that results in a val-met substitution at codon 158 results in a reduced activity level of COMT, with met-met homozygotes having a 3 to 4-fold reduction in enzyme activity relative to val-val homozygotes. The COMT-met polymorphism is unique to humans. If the frequencies of these alleles vary across populations, they may result in different types of genotypical abilities and behavioral predispositions. Indeed, the low-activity COMT, the site-present allele, MET, that encodes the low-activity enzyme, and high-activity COMT, the site-absent allele, VAL, that encodes the high-activity enzyme, alleles, show significant worldwide variation, as reported by a study surveying allele frequencies of this polymorphism in a global sample of populations, Palmatier et al. 1999. Palmatier and colleagues showed that Europeans have the highest frequency of the low-activity COMT allele, while the high-activity COMT allele is much more common in populations in all other parts of the world. COMT MET, low activity, high dopamine, allele carriers outperform VAL, high activity, low dopamine, homozygotes on a variety of cognitive tasks. However, the relationship between genotype and cognitive performance appears to reverse under stressful conditions. Stress increases prefrontal cortex dopamine levels, and MET allele carriers, with higher dopamine, show performance deficits relative to VAL allele carriers. This pattern reflects the inverted U-shaped function of dopamine activity, where too little, VAL allele, or too much, MET allele carriers under stress, dopamine is associated with poor cognitive performance. To understand the possible implications of these variations on cognitive performance, we can view the low and high COMT genetic variants as being associated with either strategist or warrior archetypes. The third, medium COMT variant, demonstrates traits from both strategist and warrior playbooks, making them a hybrid of these personalities. Though the effect of the change in COMT has been well documented in the scientific literature, the effect of one genetic variation cannot completely account for major differences in our personalities. Individual behaviors are the result of complex interactions between our genes and environment. Nonetheless, being aware of possible contributing factors to your ability to maintain focus and avoid stress is valuable to optimize your mental fitness and be your best.